Okay, I know that none of you noticed this, but this lecture got up a little bit late. Week 3, Lecture 4, Oxygen and Carbon Transfer. So oxygen is, you're familiar probably from high school and other courses. Hemoglobin and myoglobin are the storage proteins for oxygen and the transport proteins for oxygen. We're going to talk specifically about the inorganic chemistry that's going on at that heme iron. So this is in pages 81 to 87 of the book. Then carbon transfer, coenzyme B12. It's the bioorganometallic reagent. Prof. Van Hoevelen studies B12 mimics in her research, or has in the past. This is pages 87 to 92. Completely skipping electron transfer. It's important. It's part of almost every reaction that we'll discuss. We will look at electron transfer in some detail in 2 weeks when we look at the nitrogenase case study. I want to give you the take-home message on electron transfer, and that is this. Proteins carefully control the redox states of every electron carrier to precisely tune the potentials and get the most energy out of each electron. We combust our food, but instead of getting heat and light, we get carefully controlled redox events to capture the energy in reducing equivalents such as NADPH and ATP. So the slow step-by-step conversion of electrons into energy instead of a rapid combustion reaction is how life is different from burning things. So the electron transport will show up a little bit today, and we'll see it in nitrogenase, but I'm not going to lecture more on it. Okay, let's talk about oxygen. I mentioned that oxygen became available as a byproduct from photosynthesis in the last couple of lectures. And oxygen is highly toxic to life. It was highly toxic to the anaerobic life that existed in 2 billion years ago. And so life evolved to use oxygen reduction as the final step in cellular respiration. The reduction of oxygen is the final step in the electron transport chain, reducing oxygen down to water. Why is this important? Well, there are bacteria that use different final electron receptors, such as iron 3, nitrate, or sulfate. The problem is that the redox potentials for these species is much lower than the redox potential for oxygen. You get less energy out. So the redox potential of the oxygen water couple is about 0.82 volts. The iron 3 iron 2 redox couple is 0.75 volts. The nitrate to N2 reduction potential is 0.4 volts, and the sulfate to H2S reduction potential is minus 0.22 volts. So any organism that uses oxygen is getting anywhere from 0.1 to almost 1 whole extra volt of energy per unit of food that is combusted. So aerobic organisms are more efficient, and that is why life has evolved to use the oxygen that was produced from photosynthetic bacteria. In order to do that, we had to develop ways to transport oxygen safely without forming reactive oxygen species, and store it safely for the same reason. Let's first talk about myoglobin. Myoglobin was the first protein that was characterized by X-ray diffraction. You can read about that in the textbook chapter. Myoglobin is very common. It's found in most vertebrate animals. It's got a very similar structure in all of those animals. It's 8 alpha helices in what's known as a globin fold. It creates a pocket for this heme iron, and that heme iron is incorporated into the protein through hydrophobic residues that sort of lock in these hydrophobic side chains, as well as a histidine. You can see the histidine right here. This is the histidine that binds to the iron at the center of the heme. This picture shows the oxygen coordinated, and there's another histidine on this side that sometimes is not bound to anything and sometimes is helping to stabilize the adduct of oxygen. The gross structural details of hemoglobin and myoglobin are the same. Hemoglobin is approximately a tetramer of myoglobin. It's not a perfect tetramer. It's an alpha-alpha beta-beta protein. The redox potential of heme iron 3 is plus 0.7 volts, while that for oxygen is plus 0.82 volts. So oxygen is capable of oxidizing iron 2 to iron 3 when it binds. And there's a lot of good experimental evidence suggesting that that is true. So let's zoom in here and consider what would happen to the iron in the porphyrin if there was no oxygen bound. So 5-coordinate iron 2 has a weak crystal field, and we would expect it to be a high spin complex with 4 unpaired electrons. It also has a larger radius, so this iron is too big to sit inside the porphyrin plane when it's in the iron 2 state. And the homework for this week has you consider why that might be true. Upon oxidation, the iron becomes a +3 oxidation state. It adds the 6th ligand, and it becomes low spin D5, and we would expect it to have a single unpaired electron. Oxygen, when it is reduced by 1 electron, would be expected to have 1 unpaired electron. And yet when the oxygenated heme in myoglobin and hemoglobin is studied, there are no unpaired electrons. And it suggests that those electrons are paired up. The iron electron and the oxygen, reduced oxygen electron, are paired up in a antiferromagnetic coupling. We didn't talk too much about ferromagnetism in class this year, but when the electrons pair up on different centers, so an electron that pairs up with an electron here, strong coupling of those 2 electrons can actually make that a diamagnetic situation, and that's what happens in heme iron, which is shown on the next slide. Other evidence for this change in spin state upon binding to oxygen is IR. So there's a reduced bond order in the IR spectrum, and X-ray absorption, which we'll talk about next week, is one of our 3 spectroscopic techniques that we'll consider. That's consistent with an iron 3, and then the diamagnetism of the system. I wouldn't expect you to be able to reproduce these types of calculations, but I want you to at least understand what these calculations are showing you, and the textbook goes into a lot more detail. So this is the singlet state where we have paired electrons, and this was calculated at a very high level of theory to be slightly lower in energy than the triplet state, which has 2 unpaired electrons. So this is the ground state of the species, and you can see 1 unpaired electron in a dyz orbital on the iron, and we have 1 unpaired electron in the OO pi star inner orbital here. One thing I don't understand from this paper is why this isn't just a bonding interaction. It certainly looks like that you could be able to form this pi bond between the iron and the oxygen. The lobes are oriented properly, so a careful reading of the paper might give more information there. But certainly the interaction, the strong antiferromagnetic coupling between these 2 orbitals causes the electrons to be paired up, and it's consistent with only 1 unpaired electron on oxygen, only 1 unpaired electron in iron. It's an iron 3 superoxide anion. In hemoglobin and myoglobin, the binding of oxygen is selective over that of carbon monoxide. We would normally expect carbon monoxide to be a better ligand for an iron in a low oxidation state. Iron 2 isn't really a low oxidation state, but let's call it that anyway. But certainly carbon monoxide is a better ligand than O2 due to the dipole, due to the orbitals. And it used to be thought that it was a steric interaction. Carbon monoxide would bond linearly, as we've seen in the organometallics unit. It turns out the dioxygen binds at an angle of about 120 degrees, but it was thought that the CO couldn't fit here because of this histidine would block the the carbon monoxide from binding. It turns out that is no longer thought to be the case. It's of course much more complicated than that. And what happens is there's a water bound in the site that's hydrogen bonded to the histidine. That has to leave the site in order for oxygen or CO to get into the pocket. it can oxidize the iron, reduce down to the superoxide anion, and that undergoes a hydrogen bonding interaction with a protonated histidine. And so that's a stabilizing effect. Carbon monoxide doesn't get this stabilizing effect. And so it turns out it's actually stable for dioxygen to bind in this pocket by 11 kilojoules per mole. For carbon monoxide, it's destabilized by positive 3 kilojoules per mole. Okay, now let's turn our attention to hemoglobin. Hemoglobin is the protein that transports oxygen from the lungs to the cells, and myoglobin is where the oxygen is stored, usually in muscle cells where it needs to be readily accessible. So it's been known for a long time that there is a cooperative oxygen binding effect in the tetramer of hemoglobin. And the reason we know this is because of the sigmoidal shape of oxygen binding. So myoglobin is this purple curve, and as the partial pressure of oxygen goes up, the binding goes up, and then it reaches a steady state concentration of 100% saturation at a relatively low concentration, 50 torr. It turns out the concentration of oxygen in the air, the partial pressure of O2 in the air, is out here at about 150. So myoglobin is fully saturated in air. But of course, muscle tissue is not air. In muscle tissue, it could be very depleted in oxygen. We want to be able to transfer the oxygen from the blood to the muscles, so we want there to be a higher binding of oxygen in myoglobin than in hemoglobin. And this sigmoidal S-shaped curve is the reason why we know there has to be cooperative binding. The first binding of oxygen is relatively weak, but the second, third, and fourth become very strong. And out here in the air, or in your lungs, where the partial pressure is 150 torr, the binding to hemoglobin is fully saturated and actually higher than that of myoglobin. As it travels through the body to oxygen-depleted regions, the oxygen will jump to the myoglobin. There are additional effects too, mostly this pH effect. So at lower pH, the oxygen is released more readily. Where is it lower pH in your body, not counting the stomach? Well, that's in muscle cells that are being active and forming a lot of carbon dioxide. And when you have a lot of carbon dioxide, that's the conjugate base of carbonic acid. And so the pH drops as you become more acidic, and you even have a lower binding of oxygen in those cells, in those regions of those cells. So what causes this cooperative binding? And it has to do with the iron 2, iron 3 redox couple and the movement of the iron into the heme plane. When that iron moves into the plane of the porphyrin, it pulls the histidine with it, which pulls on the rest of that AABB tetramer, causing the other 3 active sites in the protein to more closely resemble that of the dioxygen binding site instead of the deoxyhemoglobin site. There's a pretty cool animation of this on the Wikipedia page for hemoglobin. It comes from X-ray diffraction data, and we'll talk about X-ray diffraction next week, but for now just think of it as a movie that shows hemoglobin bound to and not bound to oxygen, and what that does to the whole protein. A small motion of the iron and the histidine causes a really large motion in the protein due to some lever arms and some other things going on. The other thing I want to say is that hemoglobin also carries a little bit of CO2 back to the lungs. Most of the CO2 travels as bicarbonate. So remember carbonic anhydrase on the intro day from this week. That carbonic anhydrase generates bicarbonate from CO2, and it gets exhaled in the lungs. But the CO2 forms a carbamate on the terminal amines of all 4 of the monomeric forms in hemoglobin. And so that CO2 can be released back in the lungs as well. Okay, now we're going to turn our attention to biological organometallic chemistry in cobalamin. So cobalamin has a cobalt and a chlorine ring. We talked about this in the last lecture. It's essentially a porphyrin. For all practical purposes, it's a porphyrin. For the purposes of Chem 104, you can just think of it as a porphyrin, although it's slightly different from a porphyrin. These cobalamins can contain cobalt in the 1, 2, and 3 oxidation states, and it contains an axial ligand, which is a nucleotide, and there's a good picture of this in the textbook chapter. So you have a nucleotide and then an R group, and this is one of very few metal-carbon bonds found in biology. The methyl case is methylcobalamin, and that's used for methyl transfer, specifically to make methionine. There can be an adenosyl group to make adenosyl cobalamin. So this is the adenosyl group. The cobalt is bound to this CH2. This is adenosine, a DNA base. And you can also have the cyano, which is vitamin B12. And the cyano, a cyanide group, is just an artifact of the synthesis. When you take vitamin B12, you're actually ingesting cyanide. So let's think about alkylation in biology, and there's a few examples in the textbook chapter, and I'm just going to mention them briefly right now. First of all, cobalt-3 is a low-spin D6 configuration. You should expect it to be configurationally inert to ligand substitution, whereas cobalt-2 has one unpaired electron in a DZ² orbital. You can actually learn a lot about metal centers by EPR, electron paramagnetic resonance. It's essentially NMR of electrons, and we'll talk about that next week. The cobalt-carbon bond homolysis, so the homolytic cleavage of the cobalt-carbon bond is about 30 to 35 kilocalories per mole. The carbon is released as a radical. And this B12 coenzyme is just a small little protein that is recruited by other enzymes that need this radical chemistry to take place to do its other reactions. This is most often seen in breaking down of metabolites and breaking bonds and rearranging things in the Krebs cycle and other places like that. So here are two example chemical reactions. The first is the use of the adenosyl radical. The adenosyl radical, when it is released from the cobalt, it abstracts a hydrogen, an H dot, from the substrate, causing a radical to form. So this is methylmalonyl cobalt coenzyme A mutase, and you don't need to know that, but this undergoes a cyclopropanation reaction and then a rearrangement. So this has gone from a branched alkyl to a linear alkyl. And then glutamate mutase undergoes a radical rearrangement. So the first radical is formed, this part pops off as a radical, and then it adds onto the other carbon. So these skeletal rearrangements are what these radical reactions do. Finally, the H dot that's on the adenosine transfers back to the substrate, reforming the cobalt adenosyl bond. Methylcobalamin is a methyl transfer reagent that's used in the synthesis of methionine. There's evidence that the reaction occurs by an SN2 mechanism because there is inversion of configuration at a chiral carbon, a chiral methyl group, a CHDT group. However, the exact mechanism is not known with certainty. Okay, so I've alluded several times today that we're going to talk about experimental methods next week. How do we know what I've told you we know about these proteins? So EPR, so we've mentioned EPR. In fact, I've mentioned EPR once or twice today. X-ray spectroscopy, X-ray diffraction. These are all tools that the bioinorganic chemist and other inorganic chemists and other chemists use, but we're going to look at their specific use and utility in bioinorganic chemistry. And finally, Mossbauer spectroscopy, just because it's kind of a cool one that I learned a lot about when I was writing the textbook chapter, and I thought I would share that with you this coming week. All right, so take a look at the homeworks, and hopefully things are going well for you, and stay in touch.